again, sort of to track where we are with the material in the chapter, this is the stuff we just did on the innate immunity. Then we're getting into adaptive immunity, and it starts by talking about T cells specifically, and then goes into B cells. And I want to start off by talking a little bit about something that's sort of common between the two. So there's a picture that I'm going to go to in a second that's out of the B cell module, and I'll just address that to begin with, and then we'll get back just to the T cells. Now, when we're talking about adaptive immunity, we're talking about what lymphocytes do for all of this. And oftentimes it's referred to as sort of two aspects. There's cell-mediated immunity, which is what the T cells are responsible for. And different sources name it differently. Humoral immunity is sort of the one I prefer, just because I like the word humor. Not because it's funny, but because in that context, humor means bodily fluid. So humoral immunity is talking about sort of what's part of bodily fluids. And what that really means is antibodies. So antibodies are secreted from what B cells become when they're fully activated. And so the antibodies can be in the plasma, or they can be in interstitial fluid, or they can be in various secreted fluids from our body, like tears, et cetera, that sort of thing. So oftentimes they're sort of described that way. We kind of separate it out. And in the book, they start by talking about the T cells. Now, before we get in there, I actually want to talk a little bit about sort of what adaptive immunity is for and what ties the T cells and B cells together. What it's about is it's recognizing specific pathogens. So the innate response that we're just talking about is really kind of generalized. When there's something going on in your body, then you're going to have this generalized response, which will include a lot of the things we just talked about. But when you have a specific infection, like the influenza virus, the adaptive immune system has a way of targeting that specifically. And it'll target influenza virus, and it's not going to have anything to do with something else like a chickenpox virus or anything like that. Very specific. I use viruses a lot for examples of particular things because people have heard of a lot of virus-based infections, but it could be, an E. coli infection or salmonella or staph or something like that. There are a number of different types of infections. These are all very specific about what they're going to target. T cells do that through a structure that's called a T cell receptor, which is what this picture is depicting. It's a receptor, so that means that it's membrane bound. So it has a transmembrane region, the part that's actually stuck in the membrane of the T cell. And then what's sticking out is represented by these kind of four blocks. The two closest to the membrane are labeled as the constant region, and the two further away are called the variable region. So in this first part where it's talking about T cells, it sort of introduces it with this. It makes more sense to me to introduce what this kind of thing is about by going to antibodies. So this is a picture out of the second part, or the next module, which is about B cells, showing an antibody. And the structure between the two is actually fairly similar. Let me just pull this tab out for a second. Oh, come on. This never works quite the way I want it to, but okay. So I can put these side by side, and you can see that the antibody has a variable region and a constant region too. And whether it's the T cell receptor, obviously in the T cell, or the antibody in the B cell, there's genes for these different pieces. So there's an alpha chain and a beta chain gene that's active in a T cell, and there's a light chain and a heavy chain gene that's active in the B cell. Both of those genes and their respective types of lymphocytes have a region that codes for, or has a sequence that codes for the variable region in the protein that's being made. What's happening in that variable region is that the DNA rearranges itself kind of randomly. Little chunks of DNA are spliced in and out and put in random order to make for a unique type of antibody or T-cell receptor. Now, in these pictures, the one of the antibody points to the end where the light chain and the heavy chain come together in the variable region. It says antigen binding site. There's an equivalent sort of thing going on in the T-cell receptor. It's just not highlighted in this picture. But in that end part, where it says alpha chain and beta chain above it, there's going to be some inherent shape to the protein, which makes it possible to bind to something. Okay, and I'll show you a picture in a second that kind of highlights that. The issue, however, with the variable region in whichever type of molecule that we're talking about is that because of the random sorting of the DNA, which happens at the beginning of that lymphocyte's life cycle, when it's first made, it resorts the DNA, and the gene then stays constant for the rest of the cell's life. So all of the lymphocyte makes its antibody or T-cell receptor, whichever type we're talking about, that's unique compared to all of the other lymphocytes out there. But for the rest of that cell's existence, it's going to make the exact same antibody or T-cell receptor. And what's unique is what the antigen binding site part looks like out here, which it's not really depicted in the T-cell receptor here, but it's the same sort of idea. So this is a picture, again, out of the T-cell part that's depicting how the T-cell's receptors interact with an antigen. And an antigen is just whatever these things bind to. The word antigen actually means something that generates an antibody response. But it applies to T-cell receptors too. We just don't call them T-cell receptor gens or something like that, but it's the same idea. Where the T-cell receptor, which is what's in this picture, or an antibody, actually binds to the antigen itself, it's binding because of the specific shape of that thing. So this depicts what would be a protein. And so this little section of the protein, the amino acids that make up this little curve right here, have a very unique shape just because of whatever amino acids they happen to be, and because of the particular three-dimensional shape that the protein's taking on in that spot. And that's what that particular T-cell receptor interacts with, and antibodies work the same way. Now, this is a little bit of a strange picture because really T-cells interact with mainly cellular antigens, oftentimes cells from our body. Antibodies are more likely going to interact with a molecular antigen like this, but for whatever reason, this is how the book is depicting. So that part of the protein in this picture where the T-cell receptor is attached, or an antibody would attach, is called the antigenic determinant. It's the specific shape of the antigen that matches, like a puzzle piece, the antigen binding site that's in the variable region of whichever protein we're talking about. That's how these things can be specific. And in this picture, they're showing us 3 different T cells, and they each have a unique T-cell receptor. And the variable region in each one is colored differently to kind of highlight that they are unique compared to each other. And so this T-cell receptor binds to that particular part of this antigen. This T-cell receptor binds to that particular part, and this T-cell receptor binds to this particular part. We could depict the exact same thing with antibodies. It's the exact same concept. One difference, however, with antibodies is that antibodies actually have 2 antigen binding sites. So the T-cell receptor is kind of like one side of the antibody. It only binds to one thing. But antibodies have 2 identical antigen binding sites. Now, actually, in this picture, they didn't draw them identical, which is kind of a mistake. But there's 2 copies of the light chain and 2 copies of the heavy chain. They happen to be colored 4 different colors, but actually, this piece of protein that's yellow and this piece of protein that's green are just 2 copies of the exact same thing. And then the one that's colored red and the one that's colored blue, again, are just 2 copies of the exact same thing. So there's this sort of bilateral symmetry to antibodies. And at the antigen binding site at either side, they're identical antigen binding sites. They're going to bind to 2 of whatever that antigen is. So if this picture with the T cell receptors and the protein antigen were showing us an antibody, we'd have an antibody attached there. The other half of the antibody could attach to a second copy of this protein antigen and link them together. Okay. So antibodies have that ability to cross-link 2 separate antigens together. When you're infected by a virus or a bacterium, or if this is a snake venom toxin or something like that, you're going to have more than one of those things in your body. You're going to have a whole population of bacteria. You're going to have a whole mess of viruses. You're going to have a large concentration of the snake venom toxin. And so the antibodies can actually link them together, which can play a role in how we get rid of those things. But that's, whether it's T cell receptors or antibodies, that's the basis of the specificity in the adaptive immune response. Now, I want to concentrate on T cells for a little while. And what's important about T cells is because they have these T cell receptors, or well, not necessarily because they have the T cell receptors, but through the T cell receptors, they interact with antigens that are presented to them on what we would call antigen-presenting cells. In the book, they use as their sort of go-to example of an antigen-presenting cell what's supposed to be a dendritic cell. Okay. Now, partly for the sake of the textbook, I know that they do these sorts of things to set themselves apart from other textbooks. And pretty much every other textbook is going to use a macrophage as the example antigen-presenting cell. It's fine that they're using a dendritic cell. It's just not the most common example that you're going to come across. But this picture actually shows you what happens in a phagocytic cell. So here we have our sort of go-to example of a pathogen, which is probably supposed to represent a rod-shaped bacterium. It's engulfed by the cell, and it ends up inside of a vesicle, which then merges with what's called the lysosome. The lysosome is basically the stomach of the cell. It has a low pH, and it has digestive enzymes. So when the lysosome and the vesicle that contains the pathogen merge together, then the digestive enzymes and the acids in the lysosome help to digest the pathogen. And then that vesicle, which is the merged lysosome, and it's called a phagosome that contains the phagocytosed pathogen. When it's done doing its job, there are going to be little pieces of digestive pathogen floating around in there. Those end up getting bound to a molecule called MHC. Now, in this picture, they're pointing to MHC and they're calling it MHC2, which hopefully will make you think, oh, there must be an MHC1 somewhere too. And in fact, that's true. For a phagocytic cell, they use MHC2, okay, which isn't relevant just yet, but it will come up. Now, MHC actually does stand for something, and if you're really interested, you can Google it and it'll tell you. But it was named for something that it does that doesn't exactly correspond to what's going on here. It's fairly close. But over the years, MHC has been found to have a lot of functions that go beyond what its name technically means. So we're not going to refer to it by its full name. We're just going to refer to it by its abbreviation, okay, which happens sometimes. For example, when you go buy a bucket of chicken, you're buying it from KFC. That's the official name of that restaurant that you buy the bucket of chicken from. Okay? And I put air quotes around restaurant because it's fast food. But it used to be called Kentucky Fried Chicken, but they actually changed their name officially to KFC because they didn't want the word fried in their name. They figured that people realize that their name, the F in KFC stands for fried, that they wouldn't buy it. They think that it's unhealthy because, so obviously a bucket of chicken is a healthy looking meal, right? But whatever. The point I'm making is we call this MHC. That's sort of its official name as far as this class is concerned. If you're interested in what it stands for, go ahead and look it up, but we're only going to refer to it as MHC. Now, when MHC is combined with the pieces of the digestive pathogen, that is then presented on the surface of the antigen-presenting cell to a T cell. And the T cell receptor recognizes the chunk of the antigen. So this chunk of the antigen would be the antigenic determinant of whatever this original pathogen was. Okay? And this happens to be the T cell that has the T cell receptor that recognizes that specific antigenic determinant. Okay. Now, it's not labeled in this picture, but this little purple stick right here is important because when a T cell interacts with an antigen-presenting cell, it's not just the T cell receptor and the antigen that interact, but this purple stick off to the side and MHC also interact. And we'll look at that in a little bit detail later on. But I want to point it out since it's in the picture here. So this is what we mean by an antigen-presenting cell. Now, again, this is a dendritic cell, and dendritic cells are a little bit different from your average phagocyte. So they can present antigens in a different way also, which is suggested here, which I'm actually not positive if this is really what dendritic cells do, or if this is supposed to suggest what other types of antigen-presenting cells would do. Any cell in your body is going to present antigens on its surface. And the cells that do that, other cells that do that, just regular body cells besides phagocytic cells that do that, the MHC that they use is MHC1, okay, which is not exactly what this is depicting. Okay. The picture kind of shows the flow, and this could be either type of MHC, but really for any particular antigen-presenting cell, it's only going to use one type of MHC, and that'll be important for how they do their job later on. But so there are other cells that present antigens on their surface using a different MHC. In the picture here, it kind of looks like this is sort of vacuuming up little pieces of stuff from the tissue. But it could be another cell that's presenting pieces of the antigens that it regularly makes. So if it's a fibroblast, it regularly makes collagen. So it's going to present little pieces of collagen on its surface saying, here's what I'm supposed to be making. Okay. It's kind of like, if we want to go for a really extreme sort of police metaphor, if you're in a foreign country and the police stop you and ask to see your papers, okay, it's sort of the same thing here. It's like, the T cells are stopping and asking cells in your body to present their papers, to show what antigens they have. Normal cells will show normal antigens. It's diseased or infected cells that present the wrong kind of antigens, and the system will respond to that. Okay. Now, showing you the antigen-presenting cell is setting up so that we can understand how T cells become fully activated T cells, fully mature parts of the immune system. Now, T cell is just an abbreviation for the name thymocyte, which means a cell associated with the thymus. But thymocytes, or T cells, or lymphocytes of any kind, really, are all made in the bone marrow. Okay? That's where they're originally generated. T cells leave the bone marrow and go to the thymus, and that's why they're called T cells, because they go to the thymus. The lymphocytes that are made in the bone marrow but don't leave are called B cells, and the B, as far as we're concerned, stands for bone marrow. It's not actually what the B stands for, And if you take micro, you'll learn what the B stands for. But it actually is not relevant to human anatomy and physiology. So I don't ever mention what the B really stands for because it doesn't apply to this class. So we just pretend that B stands for bone marrow, although it doesn't really. So T cells, when they go to the thymus, they have to go through a couple of rounds of testing. The first round of testing is called positive selection. The T cell is tested to see if it can interact with MHC. If that happens properly, the T cell survives this round of testing and goes on for the next thing. If it doesn't interact with MHC correctly, the cell's destroyed. Because if it can't interact with MHC, a T cell isn't any good. It has to interact with MHC. Now, in these pictures, it has CD4 and CD8, minus signs next to them here and plus signs next to them here. What that's getting at is T cells end up segregating into two populations, and they're defined because they present or they express a cell surface protein called either CD8 or CD4. They've kind of put it all together in this picture, but they're ultimately going to be two different types of cells. CD8 or CD4 are those little cells off to the side, little sticks off to the side that I mentioned in this picture that are necessary to actually interact with MHC when antigen-presenting cells come along. So the way that this picture is depicting it, after positive selection, the T cells that survive that first test then start to express CD4 or CD8. I'm by no means an expert in the immune system functionality. Your micro teacher is going to have much more information than me, and it might in fact be what this picture represents, but I was always under the impression that they completely separate into two groups of cells, ones that are CD8, ones that are CD4. Now the next round of testing is called negative selection. And now the T cells are tested to see if they bind to antigens. So antigen-presenting cells present antigens, but they present self-antigens. They don't present antigens from pathogens. They present antigens that are supposed to be in your body. If they don't recognize the antigen, they survive to go on to do the next thing. If they do recognize the antigen, they're destroyed. If they do recognize the antigen, that means that they recognize a self-antigen. You don't want T cells that recognize collagen or acetylcholine receptors or something about the cell surface of any cell that's supposed to be in your body, like the A or B antigens that we talked about for blood typing. Okay, people that have type A blood are going to present the A antigen to the T cells in the negative selection stage. And the T cells that recognize the A antigen get destroyed. And T cells that don't recognize it continue on. If those T cells would recognize the B antigen, but the person doesn't have the B antigen, they're not type B blood or type AB blood, then they are immune to type B blood if they're ever exposed to it. Okay? That's why people carry that immunity to other blood types and can react with them negatively. They only get rid of the lymphocytes that recognize the antigens that are in their body, and they keep everything else. Okay. So then at the end of this, we have these T cells that have survived positive selection because they recognize MHC, and they've survived negative selection because they don't recognize any self-antigens. And then they segregate into the CD4 or CD8 populations. The CD4 positive cells go on to become what we call helper T cells, which is T subscript H. And the CD8 positive cells go on to become what we call cytotoxic T cells, which is T subscript C. That's actually not all of the types of T cells that we can get out of this, but these are the two main ones that we're interested in. Now, in the textbook, they go on to talk about the next step in another picture, and I'm not going to show you that picture just yet because that's not the story I want to tell just yet. But I do want to point out on the side of this picture, okay, we start in the bone marrow. That's where the T cells and B cells, for that matter, are generated. The T cells then leave the bone marrow and go to the thymus, which is where they go through the positive and the negative selection, and they end up becoming specifically CD4, CD8 positive cells. And then once they're at that stage, they move out of the thymus and go into other lymphatic organs. Now, what's unique about the thymus and bone marrow? How do we separate them out when we were talking about the lymphatic system last time?
Separate them out?
Yeah, why are they different? They were called something different from all the other lymphoid organs and tissues.
What to do with them being the primary?
Right. So just going back real quick to the lymphatic system part, scrolling down through this, we had the primary lymphoid organs, which is bone marrow, which is really a tissue, not an organ, and the thymus. And that corresponds actually to B cells and T cells, kind of. Okay. Everything's made in the bone marrow. The B cells stay in the bone marrow, and the T cells move on to the thymus. Those are the primary organ, lymphoid tissues and organs. Okay. After they've gotten to this point, the mature cells move from the thymus, or the mature B cells move from the bone marrow, on into other lymphatic organs. Now in the picture here, this represents a lymph node, which is the most common type of organ, but it could be the spleen or tonsils or mucosa-associated lymphatic tissue or something like that. Okay. Wherever it goes out into the secondary organs once it's mature. Now, when it's mature, oops, that's not the picture I wanted. That's the picture. When it's mature, the T cell is going to then actually interact with whatever antigen it's specific for. So I'm actually jumping ahead to a picture in the textbook for a second, because it really is more important to start the story with the helper T cell. So the helper T cell, which is the CD4 positive cell, will interact with the antigen that's presented on MHC2. Okay, CD4 interacts with MHC2, and then the T cell receptor interacts with this specific antigen. So this T cell recognizes its specific antigen presented on an antigen presenting cell with MHC2, and that activates the helper T cell. Activated helper T cells then go on to release cytokines, which will help activate other T cells and B cells. So in this picture, they go on to show you what's happening with the other type of T cell here, because this picture in the book actually comes after this picture in the book. Okay. So here we have the cytotoxic T cell. Okay. What was called the mature T cell at the end of the positive and negative selection is called a naive T cell here. It's mature, it just has never met its real antigen before. Okay. So here we have the T cell that interacts with an antigen presented to it. Just like the picture I just showed you for the helper T cell, the only difference is this is a CD8 positive T cell, and CD8 interacts with MHC1. Okay. Now, MHC1 is largely going to be used by body cells, not really dendritic cells so much, but body cells in your system And the only ones we care about, because we've eliminated any T cells that recognize self-antigens, the body cells that we care about are infected body cells. So if an epithelial cell in your respiratory tract is infected with influenza virus, it's going to start making influenza virus. And it's going to present on its surface pieces of influenza virus to the immune system. Okay. And so that's going to happen here. Okay. So let's say this is actually part of an infected body cell. That's going to lead to activation of the cytotoxic T cell. And when T cells are activated, first what they do is they make copies of themselves. They make clones, which means that every cell has the exact same DNA, down to the T cell receptor gene that's specific to this particular antigen. Okay. Some of those clones become the activated T cells that go out and destroy infected cells in your body. Other ones become what we call memory T cells that do not go out and destroy infected cells, but rather wait around in case that infection occurs again, and they're ready to fight off that infection the second time around. Now, again, in the book, they're actually showing us this picture before this picture. So this picture is kind of saying after the helper T cells doing its job, then cytotoxic T cells can do their job. What the book doesn't really talk about is what the helper T cells really do. And what they do is they help. For the helper T cell, when it gets activated, it's activated because the antigen presenting cell releases a chemical signal saying, be activated. And the antigen presenting cell that's doing that, the one that has MHC2 on it, is probably a neutrophil or a macrophage. Those are the most common antigen presenting cells that stimulate helper T cells. So those are cells that are doing a particular job. When we go to the antigen presenting cell that helps to activate a cytotoxic T cell, that's most likely going to be an infected body cell. Now, infected body cells might release a chemical that helps to activate the cytotoxic T cell directly, but that requires the infected body cell to still be able to function like a regular cell. If it's so far gone in its infection that it's no longer carrying out normal cellular functions, it might not be able to release the signal for the cytotoxic T cell to do its job. And that's where helper T cells come in. They help activate cytotoxic T cells if the antigen presenting cell can't do it itself. That's what cytokines do. Now, helper T cells will also help activate B cells, okay, which there is not a picture of in the book whatsoever. What the book has, and this is in the next section that's about B cells, but I want to address it here because it fits in with this. These are B cells that are going through the same kind of activation as we just saw. Okay. Now, there are three different B cells that are, three different naive B cells that are depicted here. And we know that they're different because the antibodies that we see on their surface are different colors. So this is a B cell that makes blue antibodies. And this is a B cell that makes red antibodies. And this is a B cell that makes purple antibodies. They're each unique antibodies. Just like if we go back to this picture here, these are three different T cells, and they all make their own individual unique T cell receptor. And the color is telling us that they're unique cells, unique receptors. Okay, so back here. In this picture, only the B cell that has the red colored antibodies recognizes this particular antigen. So there's antigens bound to its surface, it's bound to the antigen binding sites on this, but not on the purple and not on the blue ones. They don't recognize this antigen. So only this one that's activated will make clones of itself, and those clones are going to make more of the same antibody, and it'll differentiate into memory B cells and what we call plasma cells, which are the mature final form of B cells. Plasma cells look different in this picture because this sort of squiggly part in the cell represents the rough endoplasmic reticulum and Golgi apparatus, which is necessary for secreting proteins. So instead of antibodies on the surface of the newly activated, or I should say the naive B cell here, and the clonal copies of it, now the red antibodies are being secreted out of the cell into the plasma. And these are called plasma cells because that's what they do. They secrete antibodies into the plasma. So the initial activated naive B cell here divides and differentiates into plasma cells and memory B cells. That memory B cell is then going to be available to be activated by the same antigen again in the future and differentiate into more memory cells and more plasma cells to respond to a future infection. Okay. It doesn't have to go through this initial activation part back here. It just becomes more memory cells and plasma cells when it's called on. Okay. Up here at the top, the first part that involves the initial formation of the clones and all of that is the primary response. That takes a little while, maybe a couple of weeks. The secondary response, which is the memory B cell becoming plasma cells and more memory B cells immediately, is a secondary response. That happens pretty quickly over the course of possibly days, maybe even shorter, depending on the particulars of the system. And this is going to happen for every memory B cell for the rest of your life. So the secondary response isn't just the second time you see this thing. It's every subsequent exposure to that. The primary response is the first time you're exposed to something. And the secondary response is every subsequent one after that, which in the textbook they demonstrate with the B cells. But, whoops, wrong one. The memory T cells that are made, whether it's from the cytotoxic or the helper T cells, are then available to do the same thing there also. In this picture, they depict the activated cytotoxic T cells as going out and finding other infected body cells and destroying them, which they depict here in this picture and they depict here in this picture. Same idea. What's actually happening looks very different from this, but the idea is that it destroys the cell. Now, there's a very good depiction of what's happening for that in a video that I have. Now, the video I want to show you is actually from this link, but to put it on my video page in the Blackboard website, I needed some sort of YouTube video to embed there. This video is made by the same people that make the video I'm about to show you. It's a research group. I think this is just an older video showing sort of early data, and then one I'm going to show you is a later one that's a little bit more complete. So this takes us to a web page called LabRoots for some reason, and it has now embedded in it a YouTube video, which is a cytotoxic T cell moving around in, I suppose this is actual tissue, and finding infected cells and destroying them. Actually, in the video, they're showing cancerous cells, but the same could be said of an infected cell too. Now, this is a YouTube video. It just happens to not be publicly available on YouTube. Instead, it's embedded in this page. So I couldn't put this video on the video page on Blackboard. I put a link to it if you'd like to watch the whole thing. But here's a shorter one from the same people with earlier data. But I think this is a really good example. So I'm going to play this, and there's a little voiceover that explains what's going on. It actually starts with a lot of credits because a lot of people did a lot of work to do this. So we'll just go here. I don't know what Cell Dance 2014 was, but whatever. So it's a joint effort by people from Cambridge in the UK and people at NIH in Bethesda, Maryland.
These are immune cells called cytotoxic T cells. Cytotoxic T cells are able to recognize and kill virally infected and cancerous cells in the body. These T cells are on patrol, searching for their targets. Let's take a closer look at how they move. Cytotoxic T cells are constantly migrating around your body. They move through your tissues by pushing out the membrane at the leading edge of the cell and pulling themselves forward. When a T cell encounters a cancer cell, an explosion of membrane protrusions will explode the surface of the target. Cytotoxic T cells kill their targets with toxic chemicals. The chemicals are housed in these red structures called lytic granules. You can see the lytic granules polarized from the rear of the T cell to the blue cancer cell. These toxic granules secrete specifically toward the target, protecting innocent bystanders which may be nearby. As the membrane of the target is compromised by the toxins in the granules, our indicator flashes bright red, signaling the impending death of the cancerous cell. Movies like these allow us to study the efficiency with which cytotoxic T cells operate. Now we'll leave you with a few more movies of T cells doing what they do best, killing cancer.
There's such dramatic music in this. So, and then it has some credits there at the end. Now, I just want to back up and show you a couple of frames here. First off, early on, I kind of wish they had a couple of frames in here showing us what we're looking at under normal lighting conditions. What we're seeing here is a cell that's been tagged with some sort of fluorescent dye. And we're looking at this under a fluorescent scope. And so all of the black background here is just stuff that hasn't been tagged because we're just tagging the T cells. But based on the way this is moving around, or earlier how these other ones are moving around in here, there's obviously other cells in here that are not fluorescing. And it'd be kind of nice to see the context of what this is. This is some sort of culture model. It might be a piece of tissue that's been removed from an organism, and it's on a microscope, and they've tagged it with fluorescent markers, and they're shining fluorescent light on it so that they light up for the microscope or something like that. But I'd kind of like to see what else is in the neighborhood so we can kind of understand what's going on. But as this cell is moving through, the way that it moves is little bits of its cell membrane extend out, and then it pulls the rest of the cell with it. And I really like this movie because it's a really good depiction of how white blood cells move like that. And it's not just the T cells. Phagocytes move the same way. And then they switch over here to a different lighting scheme, and we've now tagged a cancer cell, and here's our T cell, and the membrane is green, and then there are these little things inside the cell which contain the chemicals that are going to destroy the cancerous cell. And there's some really beautiful pictures in here. You know, they're sort of rotating in 3 dimensions of cell so that we can see sort of the red granules inside the cell that are going to eventually be unleashed on the cell and have an effect. And so now this frame, we're looking at a slightly different scheme of coloration. We still have the green color for the T cells. Now the cancerous cells will fluoresce a very bright red very briefly when the granules that are going to kill it are released and compromise the cell inside. as I watch this, I always kind of picture like somebody inside the T cell has thrown a grenade into the cancerous cell, and you see this explosion real quick. That's kind of what it looks like to me. And so then they go through a bunch of different examples of cells doing this. Okay. Now, what's depicted here is essentially what we see in the picture here. I'm actually, let me use this, whoops, this picture to depict it. Okay. So in the book, what they use to represent the T cell is this very spherical green cell here. But this is the cell that moves all around, like we just saw in the video. And the T cell receptor on its surface, the CD protein on its surface, and the MHC on the surface of the infected cell are accentuated for the sake of this picture so we can see those. But those are going to be very, very small relative to the size of the cell. So we don't see those in the video. And then what they're pointing out here, what are called granzymes and perforins, those are the things that are released from the T cell into the target cell that's being destroyed. It's not really releasing that into the surrounding tissue at the target cell. It's really latching onto the cell and forcing those things into the target cell and making it sort of blow up. Now, it's not making it blow up and fragment exactly like this picture's depicting, but something along those lines is happening. Granzymes are enzymes that when they get into the cell, they start breaking down the macromolecules in the cell, the DNA and the proteins and all that sort of thing, and just destroys the inside of the cell. Perforins are proteins that make holes or perforate the cell membrane, okay? And so that's going to cause the cell to sort of do the same thing as complement does to bacteria that we talked about in the innate response, okay? So those are the kinds of things that do that. But really the depiction here in the picture isn't very realistic compared to what we see in the video like this, okay? The T cells latch on, and then the granzymes or the perforins are actually released directly onto the cell or into the cell and make it blow up like this, okay? So this is real data showing what's happening for these cells, and just keep that in mind based on how it's depicted in the textbooks. And textbooks always show it like this, which is, again, a little bit incorrect what's really happening. And a colleague of mine shared that video with me, the link there, and so I wanted to put it in. It came in just last week, so it was great that I could share it with everybody while I'm talking about it this week. Okay. Now, I talked a little bit about B cells, that they're activated because they're exposed to the antigen. Now, the thing about B cells is they don't need to be activated by an antigen-presenting cell. They just meet the antigen as it's floating around in the bodily fluid, wherever it happens to be, which would be the lymph in the lymph node or something like that. Okay. What B cells are going to, when B cells are activated, there's this other picture here that shows the helper T cell releasing the cytokines, which then help to activate the B cell. So this is the B cell that we see right here, okay? In this picture, we don't see the helper T cell, but helper T cells do help activate the B cells, and then they're going to clonally multiply and differentiate into the memory cells and the active cells, the plasma cells. Okay. What's important to think about for B cells is what the antibodies do. So this table kind of goes through the different types of antibodies. Now, any given B cell or plasma cell is going to make one antibody as far as what it recognizes. Just make a bunch of copies of the antibody based on what's happening out here at the end, where the antigen binding site is. But the plasma cell can make the antibodies in 5 different types. And the type is really more about what's going on at the other end of the antibody, not where it binds to the antigen, but how it interacts with other things. And these are called antibodies or immunoglobulins, which is the sort of official name for what an antibody is. We abbreviate immunoglobulin with capital I, lowercase g. And the 5 different types of immunoglobulins are just depicted or categorized by letter. So there's IgM, IgG, IgA, IgE, and IgD. Okay. Now, I want to talk about them in a particular order based on the percentage of antibodies that are found in all of your serum or all of your bodily fluids. The most common type of antibody is IgG. And this is sort of our quintessential antibody. So going back to, missed it again. This picture here, the plasma cells are releasing a bunch of copies of the antibody. These are IgGs that are being released. And they are by far the most common type of antibody. They're just antibodies that are secreted and float around in whatever bodily fluid they might be in. Then the next most common type at 13%, which is a lot less than 80% for IgGs, is IgAs. IgAs are characterized because they usually come in dimers, which means 2 antibodies are connected together. And so the IgA dimer has 4 antigen binding sites sticking off of it. Just means that it can bind to more particular antigens. They're also characterized as being secreted. And now IgGs are secreted also by plasma cells, but IgAs are secreted into mucus, tears, saliva, mother's milk, which is what colostrum is referring to, or whatever it is. So those are in secretions that can then have an effect somewhere. So mucus is going to line mucous membranes. Tears obviously are going to be on the surface of the eyeball. Saliva is in the oral cavity, as opposed to mucus, which is in the nasal cavity and other locations. And then mother's milk is secreted, obviously, from mammary glands. The thing about the mother's milk here also ties into something that's not mentioned here for IgGs, which is when somebody's first born, a newborn does not have an active adaptive immune system. Okay. So last time before I started with the podcast, I talked about immunity, innate immunity and adaptive immunity, and I talked about naturally acquired adaptive immunity and artificially induced adaptive immunity. Remember that? The naturally acquired passive immunity, meaning that it's what you can naturally get, but the antibodies are made by somebody else, is basically immunity that's conferred from the mother to the newborn. Most of that happens in placental transfer, and it's mostly IgGs that passed over to the fetus. And then when it's born, it has the mother's antibodies, and it's therefore immune to what the mother's immune to. Additionally, IgAs can be passed through mother's milk, which for the most part isn't a really big deal because the newborn already has the IgGs from placental transfer. Why IgAs in breast milk is important is if the mother's exposed to something after giving birth, but before the baby's adaptive immune system is functional, she can develop her immunity to whatever that might be, and she can pass that immunity on to the newborn through mother's milk. It's probably a very rare situation because it requires the woman to get sick right after childbirth and develop an immunity before the newborn has a functional immune system. So there's a window of maybe 6 months, maybe a couple of years where that might be relevant. And not everybody necessarily breastfeeds for that entire time, because a lot of adaptive immunity is probably online sooner than that. But it's probably a highly or a fairly rare situation that it's actually an issue to deal with. But that's a little bit more of that naturally induced passive immunity. Okay. But more importantly, IgAs are about antibodies being present in secretions at the barriers that we mentioned at the beginning with the innate immune section there. Okay. The next most common at 6% overall are IgMs. And IgMs are characterized because 5 individual antibodies will be linked together into this kind of pentameric form. Okay. This is, of course, 2-dimensional. They'd be 3-dimensional overall. But it's a little cluster of antibodies with all of the antigen binding sites on the outside for a total of 10 antigen binding sites possible. They are perfect for cross-linking cellular antigens together. So this is not a real situation where it tends to happen, but when we were talking about blood typing, clumping of red blood cells together because the A antigen reacts with A antibodies is most likely going to happen with this type of antibody. There's a bunch of places where things can bind up together, and so you make a big clump of things together. Now down at the bottom here, it says a monomer form of the IgM serves as the B cell receptor. I don't know if that's correct, and I think the book contradicts itself right away because over here, the next one to talk about at 1%, the IgD is the B cell receptor. So why they say B cell receptor here and B cell receptor here, I'm not quite sure. Again, I'm not an expert, and it might be that IgM monomers can act as B cell receptors, but I usually think of IgDs as being B cell receptors. And B cell receptors are, missed it, there we go, are the antibodies that are on the surface of B cells, okay? These are most likely IgDs, okay? They're only about 1% of the antibodies in the system because they're more likely to be necessarily at the beginning of the B cell's life cycle. And then once it's been activated and becomes plasma cells, it's going to be secreting IgGs, IgAs, and IgMs, okay, more often. Okay. So the last type is IgE, which is very, very rare, 0.002%, 2/1000th%. They might as well not exist. Okay, they're such a small fraction, but they are there. And what's important about them is that they associate with mast cells and basophils. Now, mast cells are the histamine-releasing cells of the inflammatory response that I talked about earlier. And then basophils are a very rare type of white blood cell. Mast cells cause inflammation. If there's an IgE on the surface of a mast cell, it acts as a receptor for that mast cell, and it causes the mast cell to release histamines, which is going to cause inflammation. That's often associated with allergies, okay? Because if an allergen binds to an IgE on the surface of a mast cell, it's going to cause an inflammatory response, which is not there to fight off an infection, but rather just to make your life hell, okay? Or Jasper Lawrence's life hell. Okay. Basophils are often associated, and I've never really understood exactly how, because I'm not an expert and I've not really done the legwork to figure it out. But basophils are usually associated with anti-parasitic activity, okay? Which is interesting in terms of what we learned about Jasper Lawrence. IgEs associate with the cells that are important for allergies and with the cells that are probably important for dealing with parasitic worm infestations. So that might be part of the link between hookworm and getting rid of allergies or something like that. Maybe if you have a hookworm infestation, IgEs tend towards basophils more than mast cells, so you're not going to tend to have allergic reactions. That's wild speculation on my part, but the fact that these two types of cells are the ones that are responsible for allergy and parasitic infestations, and they're related together by IgEs, suggests there might be something at play there. I don't know if it's true. Okay. So those are the different types of antibodies that a lymphocyte can make, okay, a B cell can make. And which type it secretes depends on what use the antibodies are going to be put to, which is mostly listed down at the bottom. Okay. Back to the book. Okay. Now, as it's talking about B cells and it describes antibodies and the classes of antibodies, and then it describes about B cell activation, it goes on from here to talk about primary versus secondary responses, which I talked about a little bit here, that the primary response involves the clonal selection and all that kind of thing, and the secondary response is really quick response. That's quantified in this picture here. So the primary response, measured by the amount of antibodies present in the serum over time, the primary response is pretty minor, and the secondary response is pretty major. Now, in books that depict this, most of the time on the y-axis, they just say concentration of antibody. What they don't make a point of saying, which is true, the scale on the y-axis of this figure is logarithmic. It's not linear, which means that if we had tick marks going up the side of this, it wouldn't just be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 up the side. It would be 1, 10, 100, 1,000, 10,000 up the side. Okay. The difference between a primary and a secondary immune response based on the concentration of antibodies in the blood is really like the difference between, let's say, 10 antibodies and 10,000 antibodies. Okay, it's a huge difference, which is compressed in this figure because the y-axis, again, is not linear. It's logarithmic. I don't really need to get into the math part of this. I'm just trying to illustrate the idea that the primary response is really weak compared to the secondary response. And when you're first exposed to something, it's going to take you a couple of weeks to get over the symptomology because you have this weak response and your system's really trying to figure out how to deal with it. After the second response, you get to an equivalent amount of response very quickly, and you have orders of magnitude of response left to go. So you're just going to completely overwhelm that secondary exposure and every subsequent exposure after that. The secondary immune response is based on memory cells doing their job. And here they're referring to it specifically for B cells, B memory cells. But the same thing can be said for T memory cells also, okay, or memory T cells, of which there's a helper version and a cytotoxic version. I talked about active and passive immunity already, which is there. I showed you this picture earlier, and that's the end of the B cell part. What's kind of unfortunate as they're talking about the antibodies here, they don't get into an important idea about how antibodies, or I should say antibody antigen complexes function. Complexes function, sure. That sounds kind of strange. I'm typing while I'm writing sometimes. But when an antibody and an antigen come together, there's a few things that can happen. A mnemonic device I like to use to describe this is Plano. Okay. Now, I used to, or actually the source I got this mnemonic from just gave plan, but I added the O on there because it's important to think about that separately. P stands for, hold on a second, let me remember the word. Precipitation, which is when small antigens fall out of solution. Okay, so I'm going to make this smaller. When something precipitates, that means in a solution, something falls out. The word precipitation we often use in reference to rain or something like that. So water vapor is in the cloud. When water vapor becomes denser and it starts to become drops of liquid water, it falls out of the sky. That's precipitation, just water molecules falling out of the gaseous phase in a cloud to be liquid water. In a solution, you can precipitate out a chemical by making it no longer soluble. Okay. For an antigen, let's say a protein antigen that's in your blood, if it binds to a bunch of antibodies, it's going to suddenly become too big of a complex to stay dissolved in plasma or whatever bodily fluid it's in. So it's going to precipitate out. That's what P stands for. L, equals lysis. Okay, lysis means destruction of cellular pathogens. This is what complement does. The membrane attack complex destroys the pathogen, and when we do that, we call that lysis, meaning destruction of the cell. A stands for agglutination. And this is where large antigens fall out of solution, okay? Or what we often refer to as clumping. Shoot, need to make that a little bit smaller. Okay. When we're talking about blood typing, clumping is when red blood cells react with antibodies against the A antigen or the B antigen or the Rh antigen. That's agglutination. So really agglutination and precipitation are kind of the same idea. When the antigen reacts with a bunch of antibodies, it's no longer in solution. Precipitation is referring to small antigens like proteins, and agglutination is referring to large antigens, meaning cellular antigens. And then N equals neutralization, which means the antigen or pathogen, most often, although not always, can no longer function the way it's supposed to. A good example of neutralization is if a virus particle gets coated in antibodies. When that happens, the virus particle can no longer infect a cell. Okay, it can't interact with its host cell, and so it's neutralized. Or kind of, I like to think of it in similar terms. If there's a cellular pathogen like a bacterium, especially if it has a flagellum, which isn't that common in bacteria that we're infected with, but if it is, if the antibodies bind to the flagellum so the flagellum can no longer rotate and the cell can no longer move, then it's neutralized. Okay. Or for a bacterium, it could just be covered in antibodies, which makes it unable to interact with its environment and cause disease. O equals opsonization, which means enhancing phagocytosis. Now, the word opsonization actually came up earlier in the book, and I didn't point it out. Where it comes up in the book isn't a great place, but it does come up. It's defined. Opsonization just means anything that binds to the surface of an antigen will help phagocytes locate and ingest them. It doesn't have to be antibodies, but antibodies can do that. Actually, complement proteins do that also. C3A or B, I can't remember which one, does the same thing to to pathogens. But there are a number of other molecules that can do the same thing. In the book, it's introduced in the innate response under early induced proteins. So there are some early induced proteins that cause opsonization, not just antibodies, but antibodies can opsonize a specific pathogen. Okay. Now, really, like I said, the original version of this that I started teaching with just had plan as the mnemonic. And really, at the end of any of these other things, whether it's precipitation, lysis, agglutination, or neutralization, the antigen can then be phagocytosed because of opsonization. But it doesn't have to do any of those other four things. It can just mark that antigen for phagocytosis, and it's destroyed by the phagocyte, and it's out of the picture. Okay. So I wanted to add that on at the end, because even though it is really the endpoint of the other four, it can happen by itself, and precipitation, lysis, agglutination, and neutralization might never actually occur. It depends on how ready the phagocytes are, how close they are to what's going on there for that to happen. Okay. So I really wish that the textbook included this approach to what's happening in antibody antigen in antibody function, because that's important. At the beginning, I talked about at the beginning of the section, I talked about the adaptive immune system. Lymphocytes are either about cellular immunity or about humoral immunity. And T cells are cellular. They are the attack cells that hunt down and destroy diseased and damaged cells in our body. The B cells are about humoral immunity, in that they release antibodies into our bodily fluids, which is what humor actually means, and mark them for these kinds of reactions. Okay, so either they're removed from solution, they're destroyed by complement through the classical complement pathway. Cellular antigens are agglutinated, and other antigens, usually pathogens, are neutralized. And then they can all, at the end of that, be opsonized and destroyed by phagocytes, or just directly destroyed by phagocytes before anything else happens. That's the end result of what, got the right one this time, what these antibodies that are being released are doing. They're finding the antigen and marking it so one of those five things can take place. And I wish the book kind of made that point, but they don't. Now, another thing about the book that I want to go back to, which I kind of skipped over, I want to tie this into the Jasper Lawrence story. So somewhere down here. So it has T cell types and their functions. And I showed you this picture before, which is really just about the helper T cells and the cytotoxic T cells. But they actually mention a third type of T cell. So helper T cells are abbreviated T subscript H. Cytotoxic T cells are abbreviated T subscript C. And then the third type, regulatory T cells, are abbreviated T subscript REG. Okay. Now, actually, in the text here, they didn't make them subscripts, but that's not terribly important. You usually see them as subscripts. Regulatory T cells are the lullaby cells. Okay. What they do is they regulate the other T cells to keep them from getting out of hand. And according to what we heard in the podcast, the suggestion is that they're activated by things like low-grade infections, so that when attack cells are doing their job on a regular basis, the regulatory T cells keep everything in check so that the attack cells, the cytotoxic T cells, don't get carried away and start destroying everything. Now, when we think of what were described as attack cells in the podcast, that seems to be most directly talking about cytotoxic T cells, because those are the cells that go out and kill other cells. That's what cytotoxic means. They're toxic to other cells. But to some degree, the natural killer cells and the phagocytes could potentially fall into that group also, especially because cytotoxic T cells can recruit natural killer cells and phagocytes to help do their job. So if you have an influenza infection in your respiratory tract, cytotoxic T cells are going to go out and try to fight off that infection. But there might not be enough cytotoxic T cells to get to the entire infection. So they can, when they get there and find themselves overwhelmed, recruit natural killer cells and phagocytes to help out. Okay? And natural killer cells actually release granzymes and perforin just like cytotoxic T cells do. So it's kind of like special forces going out onto the battlefield and recruiting the rank and file soldiers to help out to fight off the enemy, just to go with one of the military metaphors for that. So regulatory T cells might regulate more than just the cytotoxic T cells, but whatever it is, it keeps everything under in check so that it doesn't get carried away. And those non-specific ones, the natural killer cells and I suppose the phagocytes, are potentially the scariest because they're not specific. They don't have any T cell receptor telling them what they're supposed to attack and what they're not. So if they're running rampant, that could be part of where you start to have really big problems, like type 1 diabetes or Crohn's disease or any of these autoimmune disorders where your system starts to attack your body. Okay? And that might be part of what the Jasper Lawrence story was kind of getting at. Okay. Now, when I first learned about this stuff, regulatory T cells were called suppressor T cells. And so in the text here, or actually in the text here, they provide that alternate name. It's just regulatory is the more common name for them now. But if you hear about suppressor T cells, those are another name for the lullaby cells. So what we've done here is we started by talking about the lymphatic system and then the innate response, and then the T cells and the B cells of the adaptive response. Then what the textbook goes into is some specifics, sort of integrating the overall response to various types of pathogens here. And some of these are pathogens that I talked about last time, fungi, viruses, that sort of thing. And then it has a section that's about problems with the immune system. So that's going to fit in a little bit to what we talked about with autoimmune disorders that the Hookworm podcast kind of touched on. Or immunodeficiencies. This is specifically talking about an inherited type, or there could be, acquired immunodeficiency, which would be AIDS. That's what AIDS stands for, is acquired immunodeficiency syndrome. I think that's what the S is. And then there's the final section here is about immunology and transplants. Your immune system is adapted specifically to your body, okay? Blood type, whatever it might be. When you get a transplant of some sort of tissue, your body has to tolerate that. So when the transplant you're getting is blood tissue, blood transfusion, we want to make sure that you're getting the right type of blood so that it's not rejected. When you're getting the transplant of some other tissue, if it's a heart transplant or a corneal transplant or whatever it might be, immunology is important to think about how the body is going to tolerate that new tissue that's introduced. And so, bone marrow transplants, kidney transplants, especially in medical-based dramas, you hear a lot about those, trying to find the proper donor, because you want to find somebody whose immune system is most similar, and therefore you're less likely to reject, okay? Which may or may not be somebody in your family, okay? Your genetics and your family members' genetics have a lot of similarities, but there might be variation between you and your sibling or something like that, which makes them incompatible. Not always, and there's certainly plenty of dramas out there about the family that has another kid so that they have a spare for donations to the kid with leukemia or whatever. And so there's a whole lot of ethical issues about transplants and that sort of thing. And this also touches on cancer a little bit, because in cancer, the tissue has kind of gone crazy, and it has all sorts of weird interactions with the immune system. Metastasis of cancer is usually taking place through the lymphatic system. So it's sort of like, F you to the immune system. I'm going to find somewhere else to set up shop and form a new tumor. The video that we watched earlier was specifically T cells that are affecting cancer cells. So it's not, impossible for your immune system to react to cancer, but some of the cancers can get sort of really advanced and really mess up with your immune response. So there's a little bit about that in the book here too. So that's kind of what the whole chapter goes through. I'm not going to go through those last sections in any more detail. I just wanted to point them out to you. As for the material that you're being tested over, the part 1 and 2 questions are going to potentially be across all of the sections of the chapter, but part 3 and 4 questions are going to focus more on what I've highlighted in class, which is going to be the 2nd, 3rd, 4th, and 5th sections there. So the lymphatic system, innate immunity, T cells, and B cell stuff there. You will also have some questions in part 3 and 4 assignments that come from the podcast. So the link is in the Blackboard section, the lymphatic chapter, because that's the week that we did it. So if you want to go back and listen to that again, you can. If you didn't get the notes that I put up on the screen or that are up on the board here, in the YouTube channel version of that, you can get those. I don't think it's your particular class. I think it was my Thursday class where I actually got the lecture capture to successfully get the screen capture and the audio capture. But I don't think the audio quality is that great. So if trying to listen to the YouTube thing doesn't work, watch the YouTube and play the podcast. Try to get them to sync up a little bit. And you can sort of hear it and see what I'm typing on the board at the same time if you want to follow it around. But you will definitely have some questions coming from that. So that's all that I have for tonight. And I'll see everybody next week.